Sorry for the late start. Just had a couple of technical difficulties here this morning, so I think that is all beyond me. Let's get into this. We're going to talk about superbugs. So I meant to get to superbugs here last day because it's related to antibacterials, but we ran out of time, so we'll talk about that today. We're going to talk about some other antimicrobials like antivirals and antifungals, and we're going to have a little bit of time to talk about the midterm as well. So let's get into this, shall we? So superbugs are organisms. Usually we're talking about bacteria like 99% of the time, but these are microbes that are resistant to several antibiotics. So not just one, but many. So we're looking at multiple drugs and often multiple mechanisms of resistance. So when we are talking about superbugs, at least 90% of the time people are probably talking about MRSA. So we've mentioned MRSA a couple of times. MRSA stands for Methicillin Resistant Staphylococcus aureus. So I showed you the structure for methicillin last day, and I had mentioned it that it was considered one of those powerful antibiotics. And by powerful, I'm meaning that it has good broad spectrum activity, and it works when other drugs don't necessarily work. And at one point, methicillin, I think we're talking about the 1980s, so this is quite some time ago, methicillin was considered the drug of last resort. So meaning, okay, nothing else is working, don't worry, methicillin will work. Well, unfortunately, now we have methicillin-resistant organisms. And so this is a big concern. So what it usually means by the time something is methicillin-resistant, it is already resistant to other drugs. So we're slowly getting away from methicillin-resistant, and I'm starting to see more often in the literature, people are calling them multi-drug resistant, which is actually a better name because, of course, multi-drug is more reflective of what is going on. You'll also hear a lot of people call them MRSA rather than MRSA, just another cute way to try to say it. I don't really like MRSA so much myself, but it's used. I think you're just, when you're adding weird vowels in there, anybody can pronounce it anyway, but that's just my personal preference. So I know some people are looking at the notes and wondering, what on earth is that thing? Well, that is MRSA. I usually bring my little stuffed animal to class, and I can't find it at the moment. So I thought I'd just show you a picture. And you can see that, this is a staphylococcus. So we've got little round spherical cocci-shaped cells. And it is aureus, meaning it's gold, and it's staph, meaning it's clustered. And you'll maybe notice that this little guy has a cape, right? So this is a superbug. So of course he has a cape. And anyway, so this is one of my cuter stuffed animals that I have. I have a few stuffed bacteria I usually try to remember to bring to class and pull out at certain times to show everybody what else am I going to do with them. They're just fun to have in the office and fun to show the students. So a little bit about MRSA. So this is Staphylococcus. So you can imagine that this is, living on people. And right now, somewhere around a third to a quarter, somewhere in that range of all of us have Staphylococcus aureus on our skin. So there's about 50, maybe 53 students in this class, including myself. So that means probably somewhere on the range of, maybe 12 to 18 of us have Staphylococcus on us. And about anywhere, maybe a quarter, maybe half of us actually have MRSA. So we're looking at, my guess is anywhere from 5 to 10 of us have MRSA right now on our skin. And if it's just on the surface of the skin, like most Staphylococcus organisms, it's not really doing too much, causing too many problems. But if it gets in the skin, it can cause all sorts of skin infections. So you can see on the slide here, it's talking about skin boils and rashes. And if it gets into deeper tissues, it can cause all sorts of other problems. Staphylococcus is very good at invading pretty much any tissue you can possibly imagine. So we can see pneumonia, so lung infections. You can have toxic shock, so it's getting into the blood. And some of these infections can be very serious. So being a superbug, the reason why MRSA is so important is because it is acquired often in hospitals. In hospitals, you have people that are already ill or immunocompromised, or they have other complications, or you have invasive surgeries. So all these kind of things can make people extra susceptible, and of course, like, it's harder to treat. So I see somebody has a question and says, is it resistant to only penicillin derivatives? No, actually it has multiple forms of, there's actually a whole bunch of different strains out there. I think there are some strains that are resistant to tetracyclines, and some strains resistant to chloramphenicol, and so on. I'd have to look it up which is belonging to which strain, but I think they're typically MRSA is classified as an organism resistant to at least 3 different classes of antibiotics. So that's one reason why it's very scary. Like I said, very hard to treat. I had a friend, and she had a skin infection on her face. I think maybe I mentioned this before in class. And, I asked her about it just a little bit. I didn't want to pry too much, but I'm pretty sure that it was indeed an MRSA infection. And I'm not sure how many different rounds of antibiotics she went through before she got rid of it. And it was not fun, right? To have a skin infection on your face for, it must have been at least a couple of months or more. And, that's really unfortunate. Somebody's asking how it became resistant, or has it always been resistant? This is probably one of those things that we're pretty sure that this has basically evolved directly from just humans using antibiotics and overusing antibiotics. And so it's something that popped up in the 1980s. And as far as we know, it didn't exist before, but who knows? Maybe someone will come up with some new information at some point. Definitely has been propagated more by the use of antibiotics. So where are we finding this thing? Well, it's on your skin. So anywhere where people's skin have come in contact with, right? So other people, you can imagine all sorts of inanimate objects that have come in contact with skin, and particularly in a hospital. You could imagine things like doorknobs and surfaces, gowns, telephones. I guess payphones aren't so much of an issue anymore. And believe it or not, it's been found on pets and farm animals. So this is kind of crazy, just found about everywhere. I was going to say about payphones used to be a big issue. People always concerned about infections on payphones. And I was thinking they were an extinct thing, but believe it or not, in my travels this summer, I found 5 payphones. So I thought that was kind of interesting. I thought they were totally something of the past, but they are still popping up here and there. Someone is asking if it's beta hemolytic. Staphylococcus is usually, oh boy, I'm trying to remember, it's alpha or beta hemolytic. There's definitely hemolysis. Can't remember if it's beta or alpha. I think it's, oh boy, my brain is having a blip at the moment. I think it's beta, but I'm not 100% sure. Okay, so just some stats for you. These are some old numbers, but they haven't really changed, these numbers, in decades. 10 years ago, you can see we're looking at about 36,000 MRSA patients annually. Some of these are in-home patients as well, right? Or nursing homes and other places. And we're looking at, maybe 2,000 to 3,000 MRSA related deaths. It's one of those things that's hard to tell, right? Because, sometimes people die of, let's say, pneumonia. Well, then it's not recorded as an MRSA death. It's recorded as they died of pneumonia or something like that. So it's kind of hard to tell. But these are just some estimates from, I believe this is Health Canada. So we're looking at a lot of extra costs, and there's this number that's been floating around for a long time. If you stay in a hospital, there is a chance you will get a secondary infection. And the number is about something like 1 in 10 patients get a secondary infection, and the average stay is 10 days longer. And so that's not good. It's looking to a lot of extra cases. I had a student a few years ago, I remember after this lecture, I'm not sure what his circumstance was, but directly after this lecture, unfortunately, he had to go to emergency and got admitted to the hospital. And he came to me and talked to me afterwards and admitted he was a germaphobe. And he said in the hospital he was washing his hands like crazy because he was scared of getting a secondary infection. Poor guy. But it does happen. And you can imagine that, you know, different different hospitals, just due to design and practices, different hospitals may have different numbers on that. But it's about 1 in 10. As a hospital gets more full, you start to get more and more cross infections. So it's a big, big concern. Taken very seriously and can be life-threatening, particularly for people who are getting invasive infections. So we have to take MRSA very seriously. There are a ton of other superbugs out there, and you know, sometimes they're given, I'm just showing you this list here to mention that, you know, a lot of them I'm starting to see, you know, that for some reason, a lot of people love acronyms, and you can see these acronyms here are referring to all sorts of different things. So vancomycin resistant enterococcus. So sometimes the acronyms have to do with the drug that it's resistant to. So vancomycin, you know, if methicillin doesn't work, sometimes we use vancomycin, right? And so, you know, vancomycin is a dangerous drug and cause deafness. So it really is, we are getting desperate at that point. So when something is said to be vancomycin resistant, it is a serious organism. The second one, you can see extended spectrum beta-lactamase. So in that case, they're not even talking about the organism, they're talking about the mechanism of action, because this particular gene was spread amongst plasmids. I think this was a few years ago. It originated, they believed, in India, ended up in the UK and the US and all over the place after a while. So the ones in red, those are the ones we should know. That's why I have them in red. These are things that we are going to talk about or have talked about a little bit. So I mentioned that MRSA is the #1 superbug. Not always. Sometimes, once in a while, Clostridium difficile is the #1 superbug in a particular hospital. It seems to be cropping up again and again and again. 10 years ago, it was on the radar, but it seems that it's a lot worse than it was 10 years ago. It just seems to be more and more common, and I don't exactly know the reason for that. But Clostridium difficile seems to be kind of, you know, giving MRSA a run for its money. Pseudomonas aeruginosa, we're going to talk about that later on in the semester. I know it keeps popping up. It's kind of, I have most of my notes for Pseudomonas aeruginosa in topic 14. And so we'll get to that kind of a little bit later. Just know it's one of those organisms that is really good at being resistant to things. And Streptococcus pneumonia, there are some strains of Streptococcus that have multi-drug resistance. So by that definition, they are superbugs. So what about all these other like possibilities? You know, maybe drugs don't work. We can try the other things. So, you know, we mentioned that we have viruses that kill bacteria. So can we use those things? The answer is yes, we can. We're maybe not quite there yet, or maybe we're there, but just not able to use it yet. This has a long and complicated story of this particular type of therapy called phage therapy. You can read that little article there. There are newer articles. That's just one of the better ones that kind of talks about the history of phage therapy, because it turns out that in the Western world, we in the 1960s and 50s and so on, we spent a lot of time developing antibiotics. And in the Eastern world, we're talking about the Soviet Union, they actually practiced phage therapy. They used antibiotics as well, but they had a whole branch of science that would use phage therapy. So how does this work? It means somebody gets an infection. Let's say you get a gut infection, and so the scientists would go to the sewer, collect a little bit of sewage water, and you know, cultivate and find a virus in there that would kill the bacteria that was causing a person's gut infection. So it's not like you can just get these things off the shelf that easily, unfortunately. For most infections, it takes a scientist that has to go and collect a sample and try to find the phage that will kill that particular bacterial species. So it's complicated. And the other thing about it is that it's hard to define. So in Canada and the US and most of the Western world, our drug agencies, they want to know what's in something that you're giving to people. You can't just say, oh, it's a bunch of viruses that are cultivated from sewage. We don't like that here. Public Health Agency of Canada wants to know what you have in there. So there are companies working on this kind of thing. Maybe, we'll see some things in the future. I saw that there was, in the United States, somebody had made a phage that would kill listeria, or they found a phage that would kill listeria to spray on lunch meats. So I don't know whether that's being used or not. There was a really fascinating story about 2 or 3 years ago about a woman whose husband became very ill. And so she happened to be a microbiologist and, was like, I'm going to research this, and tried to help him with his infections. I can't even remember what the infection was. What I do remember is how the story ended, is that she had heard about phage therapy. And she took her husband and they flew over to Georgia. Now, I'm not talking about Georgia, the state in the US. I'm talking about the country over in Eastern Europe, the country that used to be part of the Soviet Union. And she had read that they were still doing phage therapy there in some places. So it's not to the degree that it used to be in the Soviet Union. But the interesting thing was she found a pharmacy. And this pharmacy had, for $2, you could buy this little flask, and apparently it had phage in it. And she bought a couple of those and gave them to her husband. And you know what? It cleared the infection. So I don't know what it was. I think it was something gastrointestinal. But after trying all these treatments in the States, a flight and $2 or $4 later, she found a treatment that, we're totally ignoring in the Western world. So this story keeps going on. This has been going on for a while. So maybe we're going to see something happen with it eventually, especially with organisms like Clostridium that are hard to treat using traditional antibiotics. Okay, so a couple other things to say about antibacterials. There's kind of this number floating around, and we're starting to do better. We're starting to do better, but you can see that that number says that up to half of all antibiotics prescribed are not necessary and appropriate. And it's hard to avoid, really hard to avoid. Physicians, they have patients coming in demanding antibiotics, and maybe the antibiotics will help, those kind of things. And so we're starting to see a lot of provinces and states and countries start to enact these things that some of them are law, some of them are more suggestions, and we're calling them antibiotic stewardship programs. And they're very complex. And if you can probably find the Alberta one online. I'm not going to go into all the details on this, but these are programs to try to figure out how they can improve antibiotic usage. So, for example, there are certain drugs that are last line of defense drugs, and we don't want to overuse them. So that means that we don't want to get resistance by overusing them. So what does this mean? It means that for certain prescriptions, doctors actually have to get a supervisor to sign off on it if it's a drug of last resort. Or for certain infections, it may mean in certain clinics we're going to rotate our drugs. So maybe February through April, we're going to use, we'll prescribe this particular antibiotic for pneumonia. And then maybe through, May through September, we'll use a different one. And the whole idea is to not give the organisms a chance to be able to get resistance. There's all sorts of other things going on with these programs, with these goals of trying to improve, obviously healthcare and outcomes and mostly to, reduce the kind of selection. I'm not going to go into, like I said, the details on these are really long, boring documents, and they're changing all the time in terms of best practices and those kind of things. But we are seeing some success in these programs, and and partly, like I said, sometimes they have to be not more than suggestions. Sometimes they have to be enforced because people are they're unfortunately people are lazy. You go back to old habits and you go to what you think works, right? And so when you start enforcing and tracking things and finding out knowing exactly what is prescribed, we can actually improve all these patient outcomes. So not going to say anything more about this. You'll probably talk about it more in some of your future classes, but they're kind of interesting from an academic standpoint. So there's a slogan I found on one online: stopping bugs and saving drugs. Okay, so let us switch to antivirals. So I mentioned before that there are hundreds of antibacterial drugs. I cannot keep track of them all. There are so many. I see a new one, I have to look them up. Antivirals, there's a lot less. And like I said, the big reason for this is that bacterial cells, they have their own cells, and their cells are very unique compared to humans. And when you think about that selective toxicity, there's so many different potential targets. They have a cell wall. We don't have a cell wall. Their DNA enzymes are different than our DNA enzymes. They have folic acid metabolism. We don't. They have different membranes and so on, ribosomes. Viruses use all of our stuff, right? They use our cells, they use our ribosomes, and so on. So there's only so many things we can do to tackle a virus. So I kind of have these divided into really, you can see kind of two categories. So why two categories? I think the textbook has 4 categories, but really you can think of it as 2 categories. You've got the nucleoside or nucleotide analogues, and I'll show you what that means in a minute. And then you've got drugs that target other viral enzymes. So these are all drugs that target some sort of viral enzyme, and that's really the bottom line here. And because that's going to be unique to the virus, and hopefully unique enough from the human versions that it's not that the drugs are not going to affect the humans. So what is a nucleoside or nucleotide analog? This means this is a chemical compound that looks like a nucleoside or nucleotide. So if you take a look at this diagram here, I have right here, this is guanosine, okay? So you may remember the genetic code. We've got A, G, T, and C, okay? So this is the G, and that's what it looks like. And we have this drug here, acyclovir. So acyclovir, if you take a look at the pink part here, so right here, it's different chemical structure than found in guanosine. But it's close enough. It's close enough that viral enzymes will bind this. Not all viral enzymes, but some, for some viruses, they will bind this, and then they make mistakes when they're trying to replicate their genetic code, or when they're trying to make, in this case here, DNA, right? And so for some reason, again, it probably has to do with the chemical mechanisms, it's binding the viral enzymes really well and not the human enzymes very well. So this is a very commonly prescribed antiviral drug. This one's been around for a while. You can see from the notes there, it's used mostly for herpes virus infections. And it comes by all sorts of trade names. You can see there's a few that I found on the internet. So Herpex and Zovirax, and there's a bunch that are off there because that are out there. Different companies have different names, and sometimes they come up with interesting names, and sometimes the names are just a bunch of weird letters mashed together. Something that I used to wonder a long time ago was where they come up with these names, and it turns out that the names are really are in many cases random letters. So when you're coming up with a name for a new drug, you can't you don't want something that means anything in any language. Like, you don't want to come up with a word and then find out in Spanish it means death or something like that. So that's one thing they want to do. They want to come up with a word, and they're going to go through all the databases and make sure it doesn't resemble something in a whole bunch of languages, right? So that's really a big part of it. And so they have to propose some names, and eventually one of those names will get approved. So sometimes these things will have a common name. So acyclovir, right? This is the name the official name, and then the companies will come up with their own names. You can see Herpex. That's actually not too bad because it actually gives you an idea that this could be used against herpes, right? Sometimes they come up with things that are suggestive, like you may have heard of Viagra, right? Viagra is being used to treat people with some sexual dysfunctions, and so the word in English, at least, it almost sounds like vital or vibrant or something like that. So it's kind of giving you the suggestion that it's going to improve your life somehow. I'll tell you a quick story about this. So a few years ago, one of my sons had these warts, and they were kind of between his toes, right? And so you go in, and because they're between the toes keep rubbing against one another, and the warts keep spreading. And it was actually, it was pretty nasty looking. So we went in and got the foot doctor. He put acid on it and burnt off the warts. And I think my son had 2 treatments, and it was not a good thing for him. He was limping for like 3 days after getting the treatment and all that, and they still just kept coming back because of those toes rubbing against one another. And then so my wife goes for the 3rd time and comes back with my son, and she's like, I'm really embarrassed. I went to the pharmacy and look what they gave me. They gave me herpes medication, right? And I'm looking at that. I said, no, don't worry. That's, I mean, this is just an antiviral. It's actually used for quite a few different, viral infections. And this is, for the warts. Warts are a viral infection, right? And actually it worked really well. I think, putting the treatment on, I think the warts were gone in like a week and a half, which, he had to live with for, I can't remember how long he had these things between his toes. So the antiviral worked really well. Kind of wish he had given that right at the beginning. So like I said, acyclovir is used for a number of different viruses, DNA viruses usually. And because it's inhibiting a DNA polymerase, I think there are some viruses that it will inhibit the RNA polymerase. But herpes viruses are pretty common, cold sores and genital herpes. So that's one of the main treatments for, or the main uses for this drug. So there are other nucleoside, nucleotide analogs out there. And the other big group, and this is a massive group, meaning in antivirals, we're talking about maybe a couple dozen drugs, are the anti-HIV drugs. So this here drug, AZT, azithromycin, was a game changer. So this is about maybe 1990 or something like that. So you can imagine in the 1980s, HIV was a thing and there was no treatment. There was nothing at all. So getting HIV was a death sentence until this drug appeared. You can see it looks a lot like thymidine, right? So, oops. Looks a lot like thymidine. AZT is just a little bit different. You can see again in that pink zone on the chemical structure. So this is a nucleoside analog. And in this case here, it is binding the reverse transcriptase. So this is binding an RNA polymerase, for an RNA virus, right? So this, like I said, was a real game changer. And so now there's people that have had HIV status, and they've been living for decades. This is really incredible. So here's the mechanism of action, right? You've got the viral RNA. Remember that with the viral RNA, we have the reverse transcriptase, and the reverse transcriptase is going to take the RNA, and eventually it's going to make a double-stranded DNA. So the drug gets in there and entirely inhibits the reverse transcriptase, so the virus can't replicate. So this can keep HIV virus levels so low in the blood of the person with HIV that it's not detectable. So this is really, really quite revolutionary. So when this came out, like I said, lots of people talking about this. The original drug here, AZT, was not the best. People got quite ill from it with side effects. They had to take quite a number of pills, and they had to take them at precise times, getting up in the middle of the night. But like I said, it was revolutionary at the time, and lots of people were talking about it. And so, of course, it inspired other chemists to say, hey, maybe we can make other nucleoside and nucleotide analogs to treat HIV. And so here's another one, and I can't keep track of these either. There's like a couple of dozen of them now, and with more mild side effects and better uses. And some of these, like I said, AZT, I think you had to have it on either an empty stomach or a full stomach, and didn't agree with certain foods. And some of these newer drugs are a little bit better tolerating those kind of conditions. Here's another one, didanosine, and this one here, like I said, is one of the newer drugs. And so AZT itself isn't quite used as much anymore because we do have do better ones. You can see this one looks a lot like adenosine. So kind of the same strategy here. These nucleoside, nucleotide analogs are starting to be used against some other viruses. And when we get to flu, I'll talk about why they're maybe not quite as effective in a moment here. So I did want to mention, this is not in your notes. I just actually made this slide this morning. And I meant to add this quite a long time ago. I wanted to talk about remdesivir. And incidentally, this popped up in the news about 2 days ago. There was a study done by the World Health Organization on remdesivir, talking about its use for treating COVID patients, right? So this is a drug that was originally developed, I think it is like brand new, meaning like maybe less than 5 years old. And it was approved to use in humans as a drug to treat Ebola. So it turned out that it was not that effective against Ebola. Other drugs were better, but we did have a drug that was approved to use in humans. And so we have a new virus this year, and so one of the first things that we did is we said, okay, let's take a look at all the antiviral drugs we have, and we'll test them in the lab, and if that works, then we'll test them in humans. So that's what happened. This was tested in the lab, and it seemed to have some activity against SARS-CoV-2 in the lab. And so now there's been, I think, about 3 clinical trials on this. And incidentally, this is one of these things where the first clinical trial came out and said that we think it's effective, but may not be statistically significant. So that's not encouraging. The second trial that came out said, hey, this is working. And then the one that was published 2 days ago said, hey, it doesn't look like it works. So what is going on here? First, let me tell you about how it works, and then I'll tell you what I think is going on. So we're often referring to this here as a prodrug. And what do I mean by a prodrug? Meaning that this is what would be, the remdesivir is what would be found in the drug that you're buying from the company. Now this is taken intravenously, it's not taken orally. So it'd be given to someone intravenously, and in the body it gets transformed into the actual active molecule. So the prodrug is just kind of a precursor, right, that gets transformed by the body. So then you get this adenosine analog here, and I don't know why it has that fancy name, GS441524. I have not seen that before. It's probably just something from the company that manufactures remdesivir. That's what they decided to call it, and it's stuck in the literature, and that's the active molecule. So this is an adenosine analog, so there's no surprise here that this is probably some sort of polymerase inhibitor. So here's the polymerase right here in SARS-CoV-2. So SARS-CoV-2 you can see is entering by endocytosis, and it's making its own RNA polymerase. It's called RDRP. I'm not sure what that stands for, and that gets inhibited by this particular molecule. So we have seen this here in the lab. You can test this and show that it inhibits the virus. So what is going on in humans? Well, it turns out with a lot of antiviral drugs, turns out a lot of them are not that effective unless, the exception is unless you have a chronic infection. So notice the drugs that I talked about already. I talked about herpes infections, I talked about having warts, and I've talked about HIV, right? So those are all chronic infections where your body is continually making new viruses. And so to treat that infection, we have to somehow stop making viruses, right? Or to, in the case of HIV, even to stop transmission to other people. The natural progress of a lot of viral diseases is you get infected. There's maybe a couple of day incubation period, then you get sick, and then your body clears it. And in the case of COVID-19, a lot of the symptoms that people are having after, let's say after 5 or 7 days, a lot of those symptoms are kind of after effects. So it's your immune system that is dealing with the after effects of having the virus. So if you've had SARS-CoV-2 they're saying to, if you're positively tested, they say 10 days after symptoms, you probably don't have the virus anymore. But some people still have symptoms. They're still coughing. There's other symptoms from blood clots and things like that. And that's all just from damage that's happened to your body and immune system reactions. So an antiviral drug isn't going to do you any good anymore. So the thing with something like remdesivir, the studies seem to show if you get it early on in your infection, then you are probably, it might help, right? Now the problem with SARS-CoV-2 is it seems that most people get infected and are incubating it for 2 to 3 days before they get symptoms. So how do you give someone the drug before they get the symptoms? Because why would we give everyone a bunch of IVs, right? So this is part of the complication with this. So right now, basically, what they're doing is this is IV only, and if somebody has a serious case, you might be able to get it. It was very publicly announced that Donald Trump had received remdesivir when he was diagnosed as COVID positive. But in that case, there was a high-profile individual, and they probably had a batch sitting around in the hospital just in case he got sick. Like I said, jury's still out whether it's actually really effective, because you got to get it to someone early on while they're actually making viruses, and preferably before they even have symptoms, unfortunately. So let's talk about some other antivirals. There's a handful of them, not too many. And I've kind of grouped them. You can see in Group B, drugs that inhibit other viral enzymes. So each virus is unique. They typically have a dozen genes kind of thing. And sometimes there's something else that a scientist can find that a drug is effective against. And I have, I think, 3 examples here, maybe 2 examples. I can't remember. So one of the examples, a high-profile drug, is Tamiflu. And there's another one that has the same mechanism called Relenza. And it was high profile in 2009 when we had our last pandemic, the H1N1 pandemic. So we were talking about the flu virus a while ago, topic 6, and I had mentioned, right, we have, your flu viruses are named, and they have, for example, we have H1N1, right? So what did that H1N1 stand for? The H was hemagglutinin, and the N was neuraminidase. So what was hemagglutinin? Hemagglutinin, the H, that was a, oops, my pen is doing tricks on me. The H, the hemagglutinin, that was an enzyme found on the surface of the virus, and it helps it to get into the host cell. The neuraminidase is kind of the opposite. It helps it to get out. So you can see there's kind of a diagram here. So I was looking for a good one, and this seems to be the best one I could find. And so you can see that part of the budding process, I'm looking right here, is the last part of that budding process is that neuraminidase actually has to cleave a little peptide there to let the virus get released. So just the last part of the budding process. So Tamiflu or Relenza actually gets in there and inhibits that. So the virus kind of gets stuck at the very last stage of its infective cycle. So this is one of these things where, again, if you can stop the life cycle of the virus, at least at some stage, then maybe you can prevent it from spreading. So Tamiflu made the company Roche very rich in 2009 because of the flu pandemic. Again, this is another one of these drugs where the studies are not completely conclusive on whether it's actually helping. You've got this situation where, again, you've got not a chronic infection, but a acute infection. And so the thing is, the earlier you give it to somebody in the infection process, the more effective it's going to be. So if you can get this drug to the person, on day 1 of having the flu, or even better, before they actually have symptoms, then it's probably going to be quite effective. And that's kind of what the studies show. The longer it's waited, the longer the person's had the virus, the less likely it's going to be effective. So that's a problem, right? But this is still prescribed pretty highly. And I'll tell you the #1 reason that I think, anyway, this is just my opinion. I don't know if there's been research done on this, but from talking to physicians on this particular issue, I believe it's prescribed for 2 reasons. Primarily, 1, you have a sick patient and it might help, right? And #2, a lot of these patients are demanding antibiotics, meaning they want penicillin. But what's the point? This is not a bacterial infection. So sometimes it's better to prescribe them something because they're demanding something rather than, sending them home and saying, get your fluids and get your rest. And like I said, it might help. So that's not necessarily a bad thing. I see somebody's made a comment here about the side effects. I'm not really familiar with the side effects on this, but it sounds like it made somebody hallucinate, so that's not fun. Yeah, I wouldn't wish that on anybody. But like I said, it certainly gets a lot of press around flu season, and Roche, the company that came up with it, has been very happy. We don't see Relenza as much because Relenza, there's a bit more resistance in the viruses to Relenza. It's a little bit of an older drug. And Tamiflu, I imagine there must be some resistant strains out there, but I'm not too familiar with it. So one other antiviral I want to show you, which is important for HIV in particular, are protease inhibitors. So there's a few of these out there. This one here, Saquinavir, is one of the ones I'm familiar with, and it is inhibiting an enzyme unique to HIV, also found in hepatitis C. I don't know as much about hepatitis C. So what is this protease that is going on in HIV? It turns out HIV has something weird that it does. So most of us, meaning most organisms on the planet, when we make proteins, we kind of make them one at a time, right? We make one piece of RNA and then we make one protein, right? So HIV, for some reason, doesn't. You can see what it's doing here. It says when the virus starts reproducing, it makes one long polypeptide. And then what it does is has a protease. When the protease comes along and cuts that into smaller peptides, and those fold and make viral proteins. And then eventually it's going to assemble and make the complete virus, right? So the protease inhibitors kind of get in there and stop that last step. So again, this is kind of one of the last steps of the viral maturation process. So I believe I have a video here. I'm going to play this for you, and you know, this is a short video, and this person does explain it a lot better than I do. And so I'll play this for you, and it's a good kind of reminder of what the some parts of the HIV life cycle. So I'll play that for you right now. I'll try to play it for you right now. There we go.
So when HIV buds from a cell, it's not in a mature form. Its proteins are actually in a multi-linked chain. So protease comes in and it cleaves the proteins in several different places. So the proteins undergo a conformational change. And actually HIV becomes mature. And the life cycle goes on. So when protease is inhibited by a medication called ritonavir, in this case, is a protease inhibitor, it binds to the active site in protease. So now it can't cleave the protein. So if it can't cleave the protein, then HIV can't become mature. And the life cycle is inhibited.
Okay, so that's protease inhibitors. We will come back to HIV. We're going to talk about the immune system after the midterm. So we'll come back and kind of talk about some of those things again as a reminder of some different treatment options for HIV. Okay, I totally forgot about this. I was just going to mention about SARS-CoV-2. There are a ton of drugs we're trying to throw at it and test, and some of them are in various stages of clinical trials. And I found this nice graphic that kind of shows some of the strategies people are trying. So you can see way down there at the bottom, polymerase inhibitor right here, remdesivir, and there's another drug they're trying that. And they're also trying different strategies. I know the chloroquine was in the media quite a while ago for different reasons and probably doesn't work. And you know, this is part of the strategy, right? Is we have a new virus, and you know, like I said, if you have approved drugs already, it's a lot easier to stick them in humans right away rather than going through long, expensive clinical trials. We can go right to the last step, which is, hey, does this work against SARS-CoV-2? So, like I said, the problem with this kind of viral infection is that you know, you got to get the drugs early on. And so drugs that are actually treating the symptoms, such as steroids, are, you know, seem to be a little bit more effective. And the biggest part of the the treatment is letting the person's immune system take care of it and treating the symptoms to a degree that the person doesn't get any more ill. Okay, so a couple other drugs to talk about. We've talked about antibacterial drugs and antiviral drugs, and I just want to spend a short amount of time talking about some antifungal and some anti-protist drugs. And I think I have kind of one example for a fungal, one protist drug, and one worm drug to kind of go through. There are a few antifungal drugs out there. I'm not familiar with all of them. I know that there's not a ton of them. And it's not that, you know, we couldn't have a ton of them, but it turns out the ones we have are decently effective. And it turns out that, that fungi and yeast don't tend to mutate to the degree that you see in bacteria. So we're not getting that kind of resistance that we see from bacteria. So we're kind of happy with the ones we have. There is some research, but not quite to the same degree. So one big group of antifungal drugs are called the azoles. So this little chemical structure here I have in the corner, that is an azole. So, you know, if you take some organic chemistry, maybe recognize that as an azole group. And so these drugs, they inhibit ergostol manufacturing. So what is ergostol? Ergostol is this little lipid. It kind of looks like cholesterol, but this is a fungal cell. Cholesterol is an animal compound. Ergostol is the fungal equivalent. So it's essential for proper membrane functioning. So if you can inhibit the production of this, they can't produce proper membranes, and the membranes technically are more, I guess you could think of them as fragile, and so it'll kill growing fungal cells. Like I said, there's a whole bunch of these out here. Here's one here, fluconazole, and if you take a look, it's got these azole groups. There's a whole bunch of these that have these azole groups, and again, you've got some weird names. You got diflocan and trican, and this is an old one here, and you can see this is some capsules, and some of them are topical creams for skin infections, and there's really a whole bunch of antifungals out there, and many of them are azoles. There are some that are not azoles, but like I said, there's a few groups of antifungal drugs, and this is one for you to know for this class. Like I said, a whole bunch out there. I was Googling azole drugs. Here's another one, myconazole nitrate. So you can see that azole group again. And some different names. We've got myconite. So people are being creative, and you can see that one is apparently for athlete's foot. And the one in the top corner, myconazole for jock itch. So, you know, hopefully that doesn't require too much of an explanation where somebody has an itchy fungal infection. And there's monostat as well, and you can see this one is for treating vaginal yeast infections. So there's a whole bunch out there. So this one, somebody is asking a question, if the membrane is damaged and the microbe is susceptible to osmotic pressure. Technically, yes, but in this case, it's more, they do have a cell wall to protect them against osmotic pressure. It's more like just a leaky membrane. So you can imagine just, you know, if your skin had a bunch of holes in it, you're not doing so well. And that's kind of what's happening here, right? It's just their membranes are either leaky or they're too rigid, meaning they can't properly transport nutrients in. It kind of depends on the temperature and a few other things. So what about anti-protist drugs? There are a few drugs out there. Unfortunately, you know, protist infections, there's a number of drugs that we don't fully understand the mechanism. Here's one, for example, the quinines, and there's a few of them. You can see they have fancy names. Mefloquine is one, chloroquine is another. These two are important historically. Chloroquine was actually discovered by so the Mayans or the Aztecs against malaria, and it comes from the bark of a tree. In the Spanish, when they had you know, part of the colonial era, were like, hey, what are you doing? You have malaria and you're chewing on that bark. And so eventually somebody extracted the chemical and figured out what it is. And so this is turned into a whole bunch of anti-malarial drugs. And what we think it is doing is, so the malaria parasite is feeding on hemoglobin, which is a protein in your red blood cells. And we think it binds to hemoglobin and makes a toxic substance. but it's one of those things that, and I'm not really fully familiar with the research here, but why we don't understand the mechanism that much, considering the importance of malaria as a disease. But you know, it's one of those things that when you read the literature, you see lots of tentative words like "we think this is how it works" or "this is the proposed mechanism" and things like that. So again, not fully sure why we don't have a better understanding of it. But these are some important malarial drugs, and there have been, you know, there's resistance to these drugs, and there are a few newer malarial drugs. We could probably use more. This is one newer malarial drug. This is interesting. I'm just throwing it in here as kind of an interesting success story of a newer drug. This was actually the subject of a Nobel Prize not too long ago, maybe 5 years ago, and this drug was discovered where there was, I think they were using robots or something like that. And anyway, it was discovered by a Chinese woman, I believe, and I'd have to look that up. But the thought was, you know, this is China, and we've got like, you know, 10,000, you know, chemicals and substances part of traditional medicine. Something might work. And so she screened something like 10,000 different substances and found that something was active against the malarial parasite. And so they came up with this particular drug here. And again, I think this is also believed to have something to do with the hemoglobin and make it toxic to the parasite at a certain stage in its life cycle. Possibly, just thought of this, the reason why we don't understand these drugs as well as we'd like to is that you can't culture the malarial parasite in certain stages. We don't know how to do it, so it might be hard to study if you can't culture it. Okay, so last couple of drugs here are dealing with larger parasites, so worms and helminths, and there are a few of these, and most of them basically paralyze the worms. And so you can see this one here is talking about blocking microtubule formation. If you don't know what a microtubule is, that's fine, but it's part of, it's a dynamic structure in the cell that allows things to move around, basically. So you're blocking that and you're paralyzing the worm, and the worm, you know, can't hold on anymore. So if this was a tapeworm, you can see this is that little reference to that article I told you to check out about a guy with a tapeworm infection. The worm can't hold on anymore, and it releases itself and gets expelled in the bowels. If the worm is found somewhere else in the body, it might leave a, you know, it might just stay there or eventually get absorbed by the body, but won't be able to, you know, live anymore. And you can see my note there about, you know, if somebody has a bunch of worms, let's say they have the Ascaris, the giant intestinal roundworm, and you know, this can lead to severe intestinal blockage. So sometimes they have to be careful and not hit people with a high dose. Sometimes surgery is necessary if there's too many worms. One newer drug worth mentioning is ivermectin. Again, a very interesting chemical structure. Humans can take this, but I'm mentioning it because some people might be familiar with it if they've had worms in their pet, and this is a pretty common drug that's used nowadays. This again is a relatively newer one. I think it's only been out for 20 years or something like that, and I'm kind of seeing it everywhere. Somebody's mentioning that they give it to the cows. So the cows getting some sort of worms of some sort, I imagine. Yeah, so, but people can take this, and I'm trying to remember, there was a story I was reading about 3 years ago. I think it was some refugees from somewhere that had arrived in, I believe it was the US. And so it was a boat or a plane of refugees, and they got off and they gave them everyone a medical check, and they gave everyone an ivermectin pill and to deworm them. I don't know if this is standard practice, but they must have had reason to suspect that these people had quite a number of parasites. So I don't know the full story behind that, why that was done. Okay, so I want to finish off this topic and to just consider a few other things to discuss about drugs. So one thing obviously is that the drug has to fit the organism, right? So, you know, if you're giving someone a drug and they have a bacterial infection, you want to give them an antibacterial drug, for example, right? If you want to, if somebody has a viral infection, you want to give them an antiviral drug. Now, there are a few of these drugs that seem to have some crosstalk, right? So, for example, hydroxychloroquine, which was given a lot of press this last year as something that might treat SARS-CoV-2. That was actually originally an anti-parasite drug used to treat malaria. It's not used so much anymore because of resistance. Some people sometimes have chloroquine. Sometimes there's parasites in fish tanks. I'm trying to think of what it's called. Remember, I haven't had fish in a while, but I had it. They get these spots. What's it called? I think it's called ick or something like that. So sometimes fish get ick. Ick is a protozoal parasite, and you can get these little tablets you stick in the water, and the tablets are supposed to kill the parasite, right? And unfortunately, there was a case in the US where this suddenly popped up in the news. Hydroxychloroquine might treat SARS-CoV-2, and like I said, now it turns out the cases is probably not helping. But there were an older couple that found it, amongst their fish tank stuff, and they took the pills, and one of them got really sick and the other died. So don't self-medicate, particularly if you're taking things that are meant for your fish tank. There might be, you don't know what concentration to take, you don't know what else is in there. So here's a little table. This is not from our textbook, from a different textbook, and showing kind of the range. And you can see some of these drugs treat gram-positives only, some of them gram-negatives only, some of them are broad spectrum, and so on, right? So this is obviously one important thing to know about drugs. I think what I tried to do with this, I modified this from the textbook, and I tried to just include the ones that we talked about in our lectures, because I know there's a lot of names. So we know this chart. I'm not too concerned about all the details here in terms of whether you know if a drug can treat a gram-positive or gram-negative. But I put this down as kind of the list of the drugs that we did talk about in this lecture. So you do need to know a little bit about these drugs. And this is a hard part for writing the midterm for this kind of thing, because I know that people get overwhelmed with all these weird words. So do your best to go through these, because I will have some questions on them. Some of them I've talked about a lot more than others. Like, you better know all about the penicillins, how they work and all that, because we spent a lot of time talking about the penicillins. Ivermectin we talked about for 45 seconds. So you do want to focus on the ones that we spent more time on, for sure. How I'm going to have the kind of questions, I'm not entirely sure yet. I think last year I had a matching question, and for antibacterials, and I think I had another question, maybe a short answer question where I said, describe one antiviral and how does it work. So I would love you to know all of them and do your best, but you do want to focus on the ones that I spent more time on, for sure. Okay, hopefully that helps. Like I said, this is always hard for me to try to write these questions, because it's, I don't want to kill you with too many details, but at the same time, it's important information. So, this is kind of one of the number one things that I got to say. It's really important that people do not get antibiotics unless they need them, okay? People way too often will have a cold or a flu, and they go to a clinic and they're demanding antibiotics. This is very bad. Usually I joke with the class, I ask, should you take penicillin if you have a flu? And if you answer yes to that question, maybe you should fail the class. I hate, it's a bit of a joke, but I'm kind of serious, right? That's a very important thing to know, right? You don't take an antibacterial drug for a viral infection. So I found this chart here somewhere on the internet, and you can see it's kind of, trying to educate people to say, hey, if you have a bacterial infection, like whooping cough or strep throat or urinary tract infection, then yeah, you want to take an antibiotic, meaning an antibacterial. The other infections are viral infections, and usually, usually there's not a treatment for that other than, treating the symptoms and letting your immune system clear it. There's lots of other things to consider, and I'm sure these are types of things you will cover in future classes. All sorts of things like route of administration, right? You can see that sometimes people can take drugs orally if you have to use IV. People are not usually doing that at home without help. There's all sorts of safety and side effects. I think we mentioned things like penicillin allergies, those kind of things. There are other side effects, right? Like, some drugs can be really hard on the kidneys. So, maybe that's not good for kids or older people and those kind of things. And I also mentioned this whole idea that the drugs can disrupt your normal flora. So you can get antibiotic-induced diarrhea or maybe a yeast infection by taking too many antibiotics and those kind of things. And you can see the last note I have there is the type of drug plan the patient is on. I had this come up when I was a university student. I had an ear infection, and then it came back, and I think it came back like 3 times. And so I probably had a resistant organism in there. And, the doctor is looking at my records, and she says, okay, I'd really like you to prescribe you this other drug here, but I don't think it's covered by your student drug plan, and it costs like $100. And then she's like, wait on just a moment here. And thankfully, she actually found a free sample to give me to try, and it helped. And I was very thankful for that because, of course, my drug plan didn't cover a drug. And so, I didn't want to get prescribed to it because when you're a student, $100 is it's like a lot of money, right? I see someone's asking about, is fluid in the middle ear different from an ear infection? It's probably caused from an infection. I couldn't tell you all the details about that. I don't really know a lot about ear infections. I just know there's different types, and there's a lot of different different ear issues that people have. Sometimes people have eczema and irritations from allergies. Sometimes people are having infections. Sometimes people get swimmer's ear, which is typically a bacterial infection from, getting water in there, and some kids are extra susceptible, and I think that has a lot to do with, the shape and size and, of their ear cavity and those kind of things, which may make you more or less susceptible. But that's kind of my guess on that one. Okay, so I think that is it for this topic. I wanted to spend a little bit of time talking about the midterm, and so let's do that. I need to just pull up the midterm slides. I know I started talking about them already, so let me just kind of finish going through that. Just load that up for you. So I think we already had gone through the first few slides. So the midterm is on Tuesday, and like last time, I will be trying to check my email this weekend. If people have questions, I'll try to respond to them. Maybe not necessarily in a timely manner over the weekend, but I will be checking my email to answer any questions. And of course, there's all of Monday to get a hold of me as well. So in this case here, we're going to be using the lockdown browser again, and do make sure you do have a good connection with the lockdown browser. And I think I had mentioned before, do treat this as an in-person exam, meaning that you're not to be having extra notes and iPads or anything like that nearby. I can tell you that there have been some cases, not in this class. Somebody's asking, why did we get less time? You should get the same amount of time. It was an hour before. Did I not write down an hour? Let me see. Yeah, it was 1 hour before, and it'll be 1 hour this time. And hopefully, like last time, I'm thinking that 1 hour should be plenty of time for the test. I will say that there have been some issues, not with this class, but with other classes. And there have been some students with suspicious behavior that have been flagged, and they've had to report to the dean and answer for themselves. So just a little warning there that we've been asked to take a look for any suspicious behavior during exam taking. And I've been told to tell people, like, if we think your exam is suspicious, then you may have to defend yourself orally, meaning you're going to have an interview and you'll get asked exam questions orally and you'll have to answer them that way. Hopefully there won't be anything suspicious, and hopefully everyone will do well on the exam. It's really, I know I don't know what to say. The whole situation is unideal for everybody, and I think, you know, we'll be happy when 2020 is over and things are getting towards normal. I'll be very happy anyway. But at least the bonus is you get to take microbiology this year, so you don't get to do that every year. So this is the breakdown. This is probably what I will do. I haven't written it yet, but this is usually the format I follow. Sometimes things are tweaked a little bit, like, you know, maybe I'll have matching worth 5 points and a few extra somewhere else or something like that. But generally this is the case. I think, you know, in this case here, I was going to rejig it from last time. As I mentioned, I'm going to have one case study and one disinfection sterilization scenario. That will be the short answer. Like I said, that's kind of the plan, and I probably will not deviate it. And if I do deviate from it, it will be very, very minimal manner. You know, in terms of like multiple choice and true and false, you know, so it used to be 10 and 5. But sometimes I do 12 and 3 or something like that. But like I said, it's not going to be a big deviation from this. Fill in the blanks, as I mentioned before, some of the fill in the blanks will be either just directly fill in the blanks, and some of them will be a little bit more short answer, or like I said, I may ask you a question like, you know, name one antiviral drug and tell me how it works, right? And so I've given that one in the past. So let's talk about these topics here for a few minutes, and just let me know if you do have any questions about any particulars on this. So all of these topics were, you know, it looks like only 4 topics, but if you remember, most of these were at least a lecture and a half. So a decent amount of material we've covered, and a lot more technical than topics 1 to 6, particularly when we're looking at all these chemicals, these antimicrobials and these disinfectants. A lot of weird words to learn. So you've got your work cut out for you. And, you know, just trying to understand the nuances. And a lot of this does, you know, I'm not testing you directly in topics 1 to 6, but it does depend a little bit on the biology of topics 1 to 6. You know, so hopefully I'm not going to have anybody come to me and say, what is a virus? Because, you know, if you're at that stage, you're going to be in really big trouble. You have a lot of work to do, even more so than anybody else. At this point, you know, we should all know what a gram positive and a gram negative are, and you know, it's about working on the details of these units. So topic 7, if you remember, we were talking about culturing organisms. So we were talking about all sorts of different media, how, you know, it works. In particular, we talked about McConkey agar and blood agar. So there were lots of different types of agar. And these are the 2 that you need to know, 100%. You need to know McConkey agar, and you need to know blood agar. And you should know what these words mean here on the left, right? So McConkey agar, for example, you know, that could be a question, right? You know, McConkey agar is a differential and selective agar. What does that mean, right? So that, you know, that's the kind of questions, you know, we're looking at, right? Or it, you know, some sort of fill in the blank around that kind of thing, right? So you definitely want to know what that means. Or maybe I'll come up with a different kind of agar and I'll explain to you what it does, and you have to tell me, you know, which of these words it fits to, right? You know, so remember, I think I did that before, the chocolate agar. I described what it is, and the answer was that chocolate agar is enriched because you're adding extra stuff to it to allow certain pathogens to grow. We did touch on a few other things. We touched on culturing other organisms such as viruses, how, you know, we might do that, and some tests that involve that you need a cultured organism to implement, right? Like a catalase test. You need a living organism off a petri dish in order to run a catalase test. So here's a sample true and false question. It says an obligate anaerobe is a species that requires oxygen to live. So the answer is false. Hopefully that was an easy one. I try to warm people up and give them an easy one. Anaerobe means anaerobic, right? So an obligate anaerobe means it must be anaerobic and it's killed by oxygen. Here's a fill in the blank. Okay, so gram-negative or gram-positive bacteria can grow on MacConkey agar. So the answer is gram-negative. So remember, MacConkey agar has the crystal violet in there, and the crystal violet actually kills the gram-positives, but the gram-negatives are fine. So that's why the media itself is kind of a purplish color. A little bit of a burgundy, but close enough. So what is selective media? Selective media is where one bacteria can grow and another species cannot. And so an example of that is the MacConkey agar. So the fill in the blank questions, you know, like I said, some of them are going to be, you have to write a sentence, others are going to be one-word answers. Just take a look at the question. Topic 8, identification. So this was a big part of this topic. We were looking at microscopy and different techniques. So techniques include staining. So we talked about two important types of stains, the gram stain and the acid-fast stain. We also talked about fluorescence, which is becoming much, much more important because it is easy to use and you're getting good results. The biggest issue is expense, but like I said before, $20,000 for a microscope, it may sound like a lot of money, but if it's saving a hospital or a clinic time, then it's probably worth the money. And if you take a look at how expensive some of the other machinery is, $20,000 is not too bad. We talked about some medically relevant bacteria, so I made some flow charts of some gram-positive and gram-negatives to know. So make sure that you know a little bit about those flow charts. Again, I don't know how I'll do that. Last year, I think I had multiple choice questions around them. You know, I think I had some questions that showed photographs of gram stains and you know, asking which one was wrong or something like that. But this could easily be a matching type question as well. You know, we have a little bit of information about an organism and have to match, you know, which one to which description. So you definitely want to go through those and learn those organisms in the flow chart. I will have enough information that if you are colorblind, you cannot distinguish between purple and pink. I will have enough information that you should be able to figure it out with other clues. So maybe cell shape or some other bits of information. So if you are colorblind, don't worry, there will be other information there, as long as you can make out the shape and other information. Somebody is asking, do people generally do worse or better on this midterm? Usually the average goes up slightly from the first midterm. So I think last year my average on the first midterm was about 70% and the second midterm was 71%. So is it harder? Yeah, I would say it's a little bit harder, but I would say people are a little bit more prepared for it. You know, and who knows, maybe it has to do with timing. I certainly have had the opposite, but generally the average does go up slightly. Now, it's not true for everybody. Some people do way better. Some people do way worse, but generally on average, slightly better. And that's actually true with most of my courses. And I think it's just because people have had a chance to do a midterm and you know, like coming into things, sometimes people just don't take the first one as seriously as they should. We also talked about serological testing, so using antibodies. And there were 3 methods that we tested, and we also talked about PCR, which is DNA testing. So I think I've had at least a couple of emails asking me about, you know, which methods require culturing. So let me just go back to this previous slide here for a second, because I think that was one of the study questions where that came from. So these metabolic tests here, these usually require culturing. And because you got to have a living organism for that, right? As for these tests here, microscopy sometimes requires culturing. You can do a gram stain directly from a pus sample, let's say, but you may not get enough bacteria in there. So for culturing, the question is, you know, for microscopy, the answer is sometimes. Kind of depends on the context. Often the culturing is done anyway. So if you have strep throat, you know, and the rapid test comes back and doesn't work, they often do a throat swab anyway, and then that gets sent to a lab and gets cultured and gram stained, right? So sometimes that is done anyway. Serological testing and PCR don't require culturing. usually a serological test is from some sort of, sample from somebody's blood or saliva or, something like that. And then they're looking for antibodies or antigens in there. And PCR, the whole idea of PCR is it's so sensitive, you don't need a culture from it. In fact, PCR is often used in organisms that we can't culture. And so, for example, testing for SARS-CoV-2, we don't culture the virus. Most of the testing done is a PCR. They're looking for viral RNA. There is some serological testing now. It's not done as routine. I think they have it at the hospital. I'm not sure why they're using it in some cases, but in that cases, often you're looking for an immune response against the virus. So it would be after somebody's infected. And so they have their uses. Okay, some sample microscopy or multiple choice questions about microscopy. So I want to show you this question here because I underlined a word, right? This is the unfortunate thing about doing online exams is you can't, mark it up. When I do multiple choice, when I do them as a test, I like to cross things off and all that. And this one, you can see in this case here, I've highlighted a word. So do read the questions carefully, and key words you do want to pay attention to. So this one is talking about visualizing a live protist, okay? So remember, electron microscopy kills things, right? We are drying them, we're coating them with heavy metals, they're done, right? Acid-fast stain also kills things because we're treating them with acid and concentrated chemicals. An ELISA test, we're not looking at something. An ELISA test is a serological test. We're not looking at something under a microscope. So the only answer here is light microscopy, right? Just normal light microscopy in this case. So do read the question. Usually most of the answers are eliminatable if you think about it, but you do have to read carefully. So like I said, unfortunately you can't write on the computer screen, and I find that very helpful. I like to see things. I like to cross things off. Here's another question. It says, which of the following can be used to detect a sample of Mycobacterium tuberculosis? So we talked about the acid-fast stain. So that one is a yes. That is a classical traditional method. So that one is for sure. I also mentioned that we're starting to use a lot more fluorescence. So that one is a yes. I didn't mention the other two, but there is a PCR test for tuberculosis. There is an ELISA test for tuberculosis. They're not used as often. I'm not sure what the contexts are, but there are tests for that. So I just wanted to mention that there are some cases where we can use multiple forms of tests. Okay, so I see we're running out of time. I will try to get through these in 1 minute. I'll go 1 minute overboard. I just want to cover these questions. Microbiology technician left out the crystal violet. So no violet, so no purple, right? So again, read the question. Nothing is going to be purple. Okay, so that leaves two left. We got C and E. So knowing a little bit about the test, knowing that what we have is things are going to be pink, everything's going to be pink is the answer to this. So let me just go through quickly. I just want to see what else we have left here. So sterilization and disinfection, a whole bunch of definitions there. Autoclave, you can take a look at these questions if you want. And I mentioned there's going to be a scenario question, so you do need to know about the disinfections and the sterilization methods. And I'm going to give you a scenario, and you're going to have to describe to me what are some appropriate methods for doing what needs to be done. So pay attention to these words, because I may ask you to disinfect something, or I may ask you to sterilize something. So it's important that you are doing what is being asked. Antimicrobials, so we talked about selective toxicity and a whole bunch of drugs. And I mentioned this is a good question. Why do we not give penicillin to treat the flu? And the answer is because penicillin inhibits bacterial reproduction. It does not kill viruses. So I think that is the end I have for today. Sorry for going over time. Do send me questions. I will answer them as soon as I can get to them. I hope you enjoy this little sign. I found this on the internet. It's kind of cool. Hopefully this is not everybody getting lost, but that you are staying on the straight and narrow and studying all the right stuff. So best of luck with your studying. I hope you all do well. Like I said, feel free to send me questions and I will do my best to answer them when I get time. So have a great weekend and try to get some sleep. Have a great day.